So, as you all know, there have been some incidental reports of carriers that had different issues. So that's what actually got our interest in starting a study if there might be phenotypes in carriers of one mutation in SLC2A10 gene. So there are a few things that were observed during these ATS clinics. So some of the carriers also had some facial features that did resemble a little bit of the ones that patients have with ATS. We also observed prominent skin manifestations in some carriers, not in all of them, with very hyperextensible skin, even EDS-like skin, Ehlers-Danlos syndrome-like skin. And we actually checked all the genes for Ehlers-Danlos syndrome in these patients as well, in these carriers. And some of them also reported musculoskeletal problems with some pectus deformities, scoliosis, joint hypermobility, and increased incidence of joint pain and muscle pain. And also some have reported that weight gain might be because of carriership. So there were a few things that kind of took our attention. Also the ophthalmological findings. So we had a few carriers that had specific keratometry done by David Warner. And one of these carriers developed keratectasia following a kind of surgery for myopia, which is actually a known complication, but relatively rare. So we just evaluated 3 carriers for that, and one of them developed keratectasia. Also, a few of the carriers also had some tortuosity of the retinal arteries. And so this has been published before. So we also looked specifically at cardiovascular findings in a few carriers. And so we noted that 1 of 9 carriers had a rather broad aorta, a disease score of 2.5, which is not yet fully pathologically, but it's at the border, upper border of normal. Mitral valve, not really prolapse, but the bulging of the mitral valve was present in 3 out of 9. And then 3 out of 9 patients had a very narrow aortic arch at -3 standard deviations, which is a remarkable finding. So we were now left with a lot of questions. Can there be any outward symptoms, pain? Is there a vascular risk for carriers? Is there a risk for eyes in carriers and so forth? So how can we tackle these questions? Well, we can do association studies. So we can compare a control population with a carrier population and then see if we find some symptoms more frequently in the carrier population than in the control population. But of course, it's important to have symptoms that can somehow be objectified. So one of the things we decided to work on first is aortic root dilatation, because you can just easily measure it. So we have a lot of people contacting us. Well, I'm a carrier for one known mutation in SLC2A10 gene, and I have these and these and these issues. But it would be wrong just to pick out just the ones with issues and a mutation, because then there are so many people in the world and so many carriers in the world. And of course, just by coincidence, a lot of these carriers will also have some of the reported issues. So actually, this is not the way how we can confirm a potential association. So what we can do is we can look at a control population, how many carriers are in a control population, and we can look in a population with aortic root dilatation. And then we could see if there are more carriers in a population with aortic root dilatation than in a control population. So how can we have an idea about carriers in the control population? Well, we have this very extensive genetic database of control people, where you can look at their full exome. And this is the GNOMAD database. So these are over 62,000 control people in this database. And here you see the data that I pulled out for the SLC2A10 gene. So these are all the variants that have been reported in this database, and the one in red are known pathogenic variants. The blue ones are the known benign variants, and then there are a lot of variants of unknown significance. Another thing you can see here in the database is if there is some kind of constraint for loss of function mutations in the gene. So are there any mutations in this gene that cause a complete absence of the protein? And if you compare then the number observed with the number of expected variants, you don't see any difference. So this is the first indication that actually this gene is quite tolerant to loss of function, meaning that having one deleterious copy of the gene wouldn't harm too much. But of course, this is only generally speaking, and maybe there might be mild phenotypes that could be that do not cause premature illness, so that these patients are still being in this control population. So another problem is what is actually a pathogenic mutation. So I've talked in the first presentation about these variants of unknown significance. So if we want to compare if there are more variants in the patients with aortic root dilatation than in the control population, we have to know at which variants we will look. So we started first at looking at some prediction scores for pathogenicity for the known mutations in the SLC2A10 gene. And we used REVEL and CAT scores for that. These are just bioinformatic tools to predict pathogenicity. And based on the scores for the known pathogenic mutations, we actually defined thresholds, being a REVEL score above 0.7, that's a scale from 0 to 1, and a CAT score above 23. And of course, we also set a population frequency. We know that carriership is relatively rare, so we could confidently say that the frequency should be below 1% for a pathogenic variant in SLC2A10. Another issue is that sometimes loss of function variants or missense variants may cause different molecular mechanisms. For instance, if you have a loss of function variant, one allele will produce a normal protein and the other allele will not produce any protein at all. And this may cause a kind of genetic compensation upregulating the normal allele that has been well known for other disorders. Well, if there is a missense mutation, it could be that there is a protein formed that is not functioning, but that is present and stable. And this will typically not cause upregulation of the normal allele. So these two different mechanisms of mutations may also result sometimes in the presence or absence of a phenotype. So we then calculated carrier frequency in the control database. And this is some basic genetic statistics. We call this the Hardy-Weinberg equilibrium. That is actually based on the assumption that the number of normal and deleterious alleles will remain stable in a certain population over time. And where you say that the wild type allele, so a normal allele frequency is P, and the frequency of alleles with deleterious mutations would be Q. So when we look specifically at loss of function variants here, we see a total frequency of all loss of function variants together of about 10,000. For missense alleles, it's about 1.3 in 1,000. And so out of this, you can estimate a carrier frequency of 1 in 634. This means that the estimated ATS frequency, so people affected with arterial tortuosity syndrome, is about 1 in 1.6 million. So we then compared several cohorts of patients with aortic root dilatation. So we have our own cohort in Ghent that we screened. We had 2 cohorts from Diana Milovic in Houston, Texas. We had the Paris cohort from Kadri Moillo, the Antwerp cohort from Bart Lewis, and then we are still awaiting data from Rotterdam. So we found a total of 9 carriers in almost 3,000 patients with aortic root dilatation without another explanation for the aortic root dilatation. And this is a p-value of about 0.05, which we call borderline statistically significant. But just adding one more carrier or one carrier less can completely change these figures. So that's making me a little bit cautious about these data. So another way to work is that we just look through carriers that are known carriers. So the obligate carriers. So we know that every ATS patient has 2 parents that are both carrier. And we could look specifically in the parents of patients to see if they have any phenotype that is occurring more frequently than in the general population. But this is quite difficult to get the data. First of all, because in most healthcare systems, they will not pay for examinations for which there is no scientific basis yet. So we are kind of in a circular reasoning. So we cannot identify a phenotype, so we cannot tell the healthcare system that these examinations should be done, so we don't identify the phenotype. So that's a bit of a problem. So we also did some functional validation studies. First of all, we also looked at electron microscopy. So once more, this is a control sample. And these are a few samples of patients with arterial tortuosity syndrome, where you can see at the borders of the elastic fibers this moth-eaten appearance with some holes, sometimes more in the inner part of the elastic fiber. If we look at carriers, we actually see quite similar picture. It's also relatively mild, but you see clear differences with a control. Next, we also looked at what we call transdifferentiation. So what we get from skin biopsies are fibroblasts. So these are the cells that produce the extracellular matrix in the skin. And if you let them grow in the lab, and a lot of these for several entities that cause vascular phenotypes. So if you look at fibroblasts with mutations in SMAD3 or TGF-beta receptor 2, they kind of transdifferentiate to a more vascular smooth muscle cell phenotype if you add TGF-beta to these cell cultures. So we did this also for carriers and patients with arterial tortuosity syndrome, and we could see that there is quite a difference with controls. So you see this, that carriers also tend to transdifferentiate faster than controls. This is also a technique where we looked at the protein levels, looking at specific markers for vascular smooth muscle cells. And you see that these are higher in carriers than in controls. A bit the similar levels as in the ATS patients themselves. And then also another test is to look at migration of these cells. So we can do a ring barrier assay where you can put the cells around a ring, and then you take off the ring, and then you see how fast they grow into the middle. And then you see that these fibroblasts from ATS patients do this faster than control fibroblasts. So we also did the same for the carriers, and we also saw that in carriers, the migration is faster than in control cells. So in conclusion, we had some indications of specific phenotypes in carriers out of the ATS clinics. However, based on our population screenings, we cannot make any definite conclusion at this point. So we would like to extend our cohorts with Rotterdam cohort, and then also with the GeneNIX data. Hopefully we can get these as well. And then another important element would be to obtain prospective data from obligate carriers where we know they are carrier, and then see if they have more specific phenotypes. So once more, I would like to thank the collaborators on this project, also the international collaborators of the referring centers, and also the lab of Jeroen Essers, who helped with some functional work on these carriers, fibroblasts. Thank you. Yeah, and of course, most importantly, we would also need you. And we thank also the carriers that were so kind to provide a skin biopsy in the past. You might find a picture for that on the ATOF website.